Maybe can I call upon Dr. Ongeni Joke
Hi, can you hear me?
from UK, Cambridge, of Chemical Engineering and Biotechnology, University of Cambridge. He will be talking on the topic generating explainable and effective data descriptor using relational learning application to cancer biology.
Hi, can you see my screen?
Yes, we can see your screen.
Okay, thank you for coming. I will be talking about some of the work we are doing in Cambridge on building descriptors for machine learning problems. So this work has just recently been published, and the paper is available online. It's open access, all the data and all the code is also available. So if you want to play around with it, it's all there. So everyone working in machine learning knows that representation or how you represent your data plays a crucial role in how well your machine learning model performs, right? So if you don't represent things properly, then your models aren't going to do very well. Even though this is not a neural net talk, it's worth pointing out that the great success we've seen of neural nets over the last 10 or so years is because of how well they do in building internal representations of the input data. So that is what we'll be talking about today, how to build representations. So in particular, we have a data set. So it's a gene expression data set from the library of integrated cellular signatures with 978 landmark genes. What this expression data is what was observed when different cancer cell lines were perturbed with certain drugs at different dosages and at different time points, right? The interesting thing with the data set is they only provide the output, they do not provide the input, right? Because it's lab-based, so it's not possible to have that. But they do provide the metadata for these responses. So they tell you what the cell line was, what the cell site was, what drug was used, and they provide the canonical smiles. They tell you what the dosage was. They also tell you at what time point was the measure taken. So our goal for this particular project was to build a representation that we can use in making predictions. What's typically done is, since we're more interested in the drugs, right, because they are what cause the sort of change we observe, is that you take the canonical smiles and you convert them to Morgan fingerprints, which are essentially binary features, and they are, you can use different sizes. It could be 124, it could be 248 bits, but that's up to you. And that produces this sort of representation for the drug, right? So for a particular bit, it could be 1, for a sample, and for another one, it could be 0. And of course, we also formalized the other metadata information. We also represented, the other metadata information as binary features as well. So we can indicate what cell line was used for a particular signature by saying 1, if that's what was used, or 0 otherwise, dosage, and so on and so forth. But what we do care about here the most is the drug and what the drug does. So we wondered if we can try something else. So we said, okay, we have the Morgan fingerprint approach, but can we improve upon this representation? And we chose to use relational learning because for such problems in the life sciences, we usually have a lot of background knowledge, which is usually not used in machine learning. So we went ahead and formalized knowledge for each of these drugs from each of these different data sources. So from Reactome, we got pathways, From drug bank, we got known drug targets, so on and so forth. And then we formalized all of this as predicates in first order logic. So we formalized all the knowledge we had for every drug as predicates in first order logic. And then we learned frequent patterns on this data, right? And these relations look something like this, which I'll talk more about in a few minutes. But we use the Warmer algorithm, and this is an inductive logic-based algorithm. There isn't enough time to discuss the details here, but if you have a look at the paper, it's pretty well described. So we learn relations, right, about the drugs in this form. Right? And what's immediately clear is that this representation is easily understandable. So for example, this simply says, well, we know that it's somewhat common, right, that a drug has a drug-gene relationship, and that gene has some function, and that function is that. And that function positively regulates some other thing. And we have more chemical-based ones where we say, well, there's a drug, it has an oxide functional group, and that drug also has a benzene ring, and there are two of them in the drug. So we then converted these relational patterns into features for our model. So as with the Morgan fingerprints, we said, well, if this relation covers a sample, then we assign a 1. If it doesn't, we assign a 0. Pretty straightforward. So simple one-hot encoding, essentially. So at the end of this, we had 2 input data sets. We had one that was based on Morgan fingerprints, and we had another one that was based on relational learning. So for our evaluation, of course, we used gene expression data from links, as I've said before. We looked at 46 cancer-related genes selected by dissimilarity. There were 1,000 of them, and it's computationally expensive to build models on. I mean, to build 1,000 several models, because you also have to do hyperparameter tuning and all of that. So we just selected 46 cancer-related genes selected by dissimilarity, and then we had 7,000 training samples and 3,000 test samples. And we considered lasso, ridge regression, and random forests. So what did we find? So when we compared fingerprints by themselves against the relational data set, we found that for all 3 of these learners, the fingerprints outperformed the relational features. Right? So you can see for lasso, for ridge regression and random forest, and this difference is indeed statistically significant using paired t-tests. And then we compared a case where we concatenated, right, the fingerprints and the relation. So we built this massive data set where we joined the fingerprints and the relations together. And then we compare that to just the relational data set. And we see that, yes, concatenating the fingerprints and relations together does outperform the relational input data set by itself. And then we compared the fingerprints and relations against just the fingerprints. And again, here we found that for all 3 of these learners, the fingerprints outperformed simply concatenating the fingerprint and the relational data sets for all 3 learners. So, well, we said, okay, instead of simply concatenating them, why don't we stack the predictions, right? So we make the predictions for the fingerprints independently, and then we make the predictions for the relational input data set independently as well, and then we stack those predictions. And we see that essentially stacking essentially wins all the time against the relational input data set for all 3 learners. And then when we compare that, so stacking against the fingerprints, right, which I think this is the most interesting result we found, is that Even though it's not as bad as before, for read regression and lasso, fingerprints still outperform stacking the results. But for random forest, the very opposite is the case, right? So if you recall, fingerprints outperformed the relational data set by itself for random forest initially, but stacking the results seems to do a lot better. So it outperforms simple fingerprints by itself on more than 40 of the 46 odd genes we considered. And then if we compare stacking to fingerprints and relations, same thing here. We find that for read regression and lasso, combining the fingerprints and relations outperforms stacking. But for random forest, stacking still outperforms combining, so simply concatenating the fingerprints and the relational data set. So, I mean, at the end of that, we want to reach a conclusion. So it's what representation should one use? And the answer is it depends. It's not really straightforward, because as our results show, it depends on the choice of learning algorithm. So what input data set works best is going to depend on the underlying learning algorithm you use. And then how the predictions are used also matter, right? So if you simply concatenate the data set, as we've seen, it doesn't work very well for some learners. But if you stack the predictions of the different input data sets, then you can do a lot better, as we've seen with random forest. So in summary, we can learn effective data descriptors using relational learning. The learned descriptors are understandable. The choice of learning algorithm and how the predictions are integrated impacts predictive performance, as you've seen. And then something I should note is that even though the descriptors themselves might be understandable, at least a lot better than standard descriptors that you usually get in QSAR-related problems, is that if you then chop those features into a hard-to-understand model, it's not really going to help you much in terms of how well your final model does, in terms of understanding your final model, sorry. So for example, if you put it in something like a decision tree, yeah, it makes sense. But if you put it in a deep neural net or an attention neural net or cognet or whatever, you're going to lose all of that understandability, even though there are efforts to make that more understandable. So in terms of current and future work, we are currently extending the relational learning algorithm to deal with very large knowledge bases. So we provide the knowledge base we use for this project. It's all online. It has something on the order of a few million facts. But the learning of the relational features was limited because the algorithm or the underlying substrates which we use to learn these features cannot deal with such large databases. And you can only constrain it to a really small portion of the knowledge. And we're also adding additional background knowledge for compounds. So for example, their 3D molecular structure, chirality, etc., etc. And with that, thank you.